Gene linkage, beyond Mendel's piece. Hello and welcome. Today we will discuss gene linkage. Have you ever wondered why certain traits seem to be passed down together over generations? It's not magic, but the fascinating phenomenon of genetic linkage. Let's dive deeper into this concept, beyond the basics, to explore its complexities and implications. Gene linkage describes the tendency of DNA sequences that are close together on a chromosome to be inherited together during the meiosis phase of sexual reproduction. Genes whose loci are nearer to each other are less likely to be separated during crossing over, and are therefore said to be genetically linked. Imagine chromosomes as highways, and genes as cities along those highways. Cities close together are more likely to be visited on the same trip, right? Similarly, linked genes tend to stick together during inheritance. This contrasts with Mendel's law of independent assortment, which states that genes for different traits are inherited independently of each other. Linkage is an exception to this rule, occurring when genes are located close enough on the same chromosome. Crossing over, also known as homologous recombination, is a crucial process during meiosis. It involves the exchange of genetic material between homologous chromosomes, leading to new combinations of alleles. Think of it as shuffling a deck of cards. Crossing over shuffles the genes on the chromosomes, potentially separating linked genes. The closer the genes are, the lower the chance of this shuffle happening between them. The frequency of crossing over between two linked genes is proportional to the distance between them. This principle is used to create genetic maps, which show the relative positions of genes on a chromosome. Recombination frequency, RF, is a measure of the proportion of recombinant offspring produced in a cross. It's calculated as the number of recombinant offspring divided by the total number of offspring. An RF of 0% indicates complete linkage, meaning the genes are so close that they are always inherited together. An RF of 50% indicates independent assortment, meaning the genes are either on different chromosomes or far apart on the same chromosome. RF values between 0% and 50% indicate varying degrees of linkage. The higher the RF, the further apart the genes are on the chromosome. Understanding gene linkage has numerous applications in genetics and related fields. It's crucial for constructing genetic maps, identifying disease genes, and predicting inheritance patterns. In agriculture, linkage analysis can help breeders select for desirable traits that are linked to other beneficial genes. This can accelerate the process of crop improvement. In medicine, identifying linked genes can help pinpoint the location of disease-causing genes. This can lead to better diagnostic tools and potential therapies. Linkage isn't always straightforward. Factors like sex-linked genes, multiple crossovers, and the presence of modifier genes can complicate the analysis. Sex-linked genes are located on the sex chromosomes, X and Y in humans. Their inheritance patterns differ between males and females due to the different chromosome compositions. Multiple crossovers can occur between two genes, potentially masking the effects of linkage. Statistical methods are used to account for these complexities. Gene linkage is a fundamental concept in genetics that explains why some traits are inherited together. Understanding linkage, crossing over, and recombination frequency is essential for graduate students in life sciences. I think this video has been informative for you. Please share it with your friends and like it. For more such information, please subscribe to this channel.